All right, Julia, you can go ahead.
Yeah, sure. Let's wait a little bit. People are still joining. Yeah. So I can see the numbers.
Julia, you can go ahead and start the.
Yep. Let me wait for a few more seconds to let others to join. I can see that people are still joining the seminar. So yeah, a few more seconds. Yeah, okay. So good morning, everyone. And that's my great privilege to introduce my longtime colleague, Dr. Andrew Byrnes at the Masonic Cancer Center Seminar Series. Dr. Byrnes received his bachelor and master degrees in molecular biophysics and biochemistry from Yale University, and then later a PhD from University of Oxford in UK. In 2000, Dr. Byrnes joined the FDA, and now he holds a position as a chief of the gene transfer and immunogenicity branch in the division of cellular and gene therapies at FDA Center for Biologics Evaluation and Research. So being with the FDA for more than 20 years, Andrew reviewed numerous IND applications and protocols and has a tremendous expertise when it comes to the GMP manufacturing of viral and non-viral therapeutics, and overall the clinical translation of gene therapies and other investigational biologics. In parallel, he's a very well-recognized scientist in the field of virology, adenovirus gene therapy, and innate immunity to viral vectors. His lab at the FDA is focused mostly on adenovirus-based vectors. That's what we do in my lab. And he's also interested in biodistribution of adenoviral vectors in rodent system and their interaction and the interaction of virions with natural antibodies and immunity. So, but what I do value most is that Andrew is a very thoughtful, deep, and I would say an old school scientist who has the fundamental knowledge in his area of expertise, and whose publication we want to read over and over again. And Andrew, that's what we did, and that's what we do. And my lab members can just only confirm that. And mostly the reason behind that is your publications is the real source of the fundamental science, the true science, and that's how we get our knowledge and the concept of adenovirus-based therapy, and not only. So for which we are entirely grateful to you. So please join me in welcoming Dr. Andrew Byrnes. And Andrew, we are looking forward to hearing your talk entitled Understanding Intravascular Delivery of Adenovirus Gene Therapy Vectors. Please.
Good morning, everyone, and thank you, Julia, for the invitation and that very kind introduction. It's a pleasure to be here, even if only virtually. I'm very sorry we couldn't be there together today, but this is the next best thing. So I'm going to start, before I get to the research from my lab, with a little bit of an introduction to the vision of cell and gene therapies, because I know you're probably curious about that. Our division regulates all of the cells and gene therapy products at the FDA. This includes a variety of genetically modified cells, including genome edited cells, a variety of vectors, including replication competent and incompetent vectors, including adenoviruses, which you'll hear about today. And then a variety of cell therapies, including differentiated cells, but also stem cell products, xenotransplantations, pig islets is starting to become a big thing again. Therapeutic vaccines for cancer treatment, and a variety of combination products, for engineered tissues, for example, and certain medical devices that are used to produce cell therapies at point of care. And the the staff here, about half of us do full-time review and the other half do review and laboratory research. We have 14 labs here and the topics span the variety of products that you see here. And we do IND review, BLA review, lots of meetings with sponsors and companies, GMP inspections of manufacturing facilities, and development of policy and guidance. And this is all getting really busy over the past 6 or 7 years. So here's the number of meetings that our office, the Office of Tissues and Advanced Therapies, has had with sponsors, investigators, and companies. And this is driven in part by a huge increase in the number of new gene therapy INDs. Although it's leveled off a little bit in the past few years, we're not sure if that's due to COVID or something else. So this is a very busy time for the whole field and for us at the FDA as well. Very exciting time as well. All right, so switching gears to my lab and our work on adenoviruses. Adenoviruses, as you know, are a large group of viruses, many of which cause colds. And the most common serotype Ad5 is mostly what I'm going to be telling you about today, and that causes a common cold. These have always been and continue to be a very popular vector in clinical trials. And they can be used to create both non-replicating vectors that transduce cells and produce a new gene and deliver a new gene and produce a new protein, or they can be designed to conditionally replicate, for example, in tumor cells and lyse those tumor cells and then spread to other cells. And you can combine this as well to express new transgenes also. And as I mentioned, this is a very popular class of vectors, although AAV vectors, adeno-associated vectors, which are completely unrelated to adenoviruses, may be getting all the press these days. There's still a very large number of clinical trials with adenoviruses, more than 90 in our division. Most of them, as I mentioned, are based on one serotype, Ad5, but this is changing and people are starting to use more serotypes. And our research is also changing to accommodate that. Almost all of these adenovirus therapies in our division are cancer therapies. And then also in a different part of our center, the Office of Vaccines Research and Review, there's more than 40 adenovirus-based vaccines for prophylaxis of infectious diseases, including a bunch of COVID vaccines naturally that you've all heard about. So the focus in our lab is systemic gene therapy with adenovirus vectors. So we deliver them intravenously into mice. The reason we use mice, we've also used rats in some studies as well, but the reason we use mice is this remains the most common preclinical model for adenovirus-based products. So it's important to understand how the vector behaves in mice and whether that's relevant to how the vector will behave in humans or not. The potential of the systemic route of delivery through the vascular system is that you could get gene delivery to any organ or tumor throughout the body. But the reality is adenovirus vectors have very poor pharmacokinetics. They're cleared very rapidly from the circulation with a half-life of just a few minutes. And in addition, these vectors often cause acute toxicity because they activate the innate immune system. And the toxicity is especially evident in the liver. And that's been the topic of a lot of our research over the past 20 years. So Ad5 vectors are hepatotropic in two senses. One is that they're very good at transducing hepatocytes. So they get inside the hepatocytes and they can be used to express proteins there. So in our experiments, we're typically going to be measuring liver transduction using luciferase enzyme activity or beta-galactosidase in some cases. And then we can also see a vast amount of clearance by these macrophages called Kupffer cells, which are located in the sinusoids in the liver. And we can detect adenovirus uptake within minutes using immunofluorescence and confocal microscopy. So a little bit more about, a little bit of background about the importance of adenovirus vector biodistribution before I get to Kupffer cells. So as I mentioned, most of the vectors cleared by the liver, and that means unless you're targeting the liver, only a small percentage reaches your destination, like your tumor that you're trying to target. And of course, hepatotoxicity can limit adenovirus gene therapy. So we need to learn how to engineer adenovirus vector tropism. Our lab has almost exclusively focused on how to detarget the vector from its normal receptors. And of course, another important aspect of that many labs are working on is retargeting the vector to other tissues or tumors. So both of these things are necessary to make adenovirus vectors more specific. And we think it's difficult to make progress if we don't understand the mechanisms. And also it's difficult to trust the preclinical models. So if you don't know how it works in mice, how can you be sure it's going to work in the same way in humans? All right, so now Kupffer cells are macrophages in the sinusoid of the liver, as I mentioned, and it was discovered by Carl von Kupffer quite a long time ago that when you inject India ink intravenously, it gets taken up by these macrophages in the sinusoids. And India ink is basically particles of carbon, agglomerates of carbon. So Kupffer cells are very good at taking up particles, including viruses, which are just particles after all. And when we inject adenovirus vectors intravenously and then use confocal microscopy to see where they end up in these Kupffer cells here, shown in red. Here's adenovirus detected with immunofluorescence, and here's the overlay. And this is 10 minutes after an IV injection into mice. And we've published a lot of work in the past on the mechanisms for this. The mechanisms include recognition by scavenger receptors. And I'm not going to tell you about that work today, but I will tell you about our work on IgM antibodies. I'll also call those natural antibodies, and the complement system. And all three of these mechanisms are relevant to uptake of these adenovirus vectors by Kupffer cells. Some of the toxic consequences of vector clearance by macrophages, and I'm just going to briefly go over three publications that we've published in the past. One is the uptake by the Kupffer cells causes their rapid death. So this happens within a few minutes of uptake. Here we're looking after 10 minutes of an adenovirus injection, and we've also injected propitium iodide intravenously. And these macrophages are all permeable to propitium iodide. They've been killed very rapidly by the adenovirus vector. We've also found that macrophages are involved in releasing a toxic lipid mediator called platelet activating factor. And this can cause shock with a high enough dose of adenovirus vector given over a short enough period of time. So here we're looking at hemoconcentration very rapidly within half an hour after administration of the vector intravenously. This is also associated with hypotension, and it can be quite serious. And then we've also found that adenovirus very rapidly activates the complement system after injecting the vector intravenously. And here again, within 30 minutes, we see accumulation of C3a, which is a breakdown product of complement, accumulation of C3a in the plasma. Now, one of the most important things to know about adenovirus vector biodistribution is that it may not depend so much on the actual receptors that the virus binds to. In fact, it depends more on the proteins in the plasma that interact with the vector. And this is all much more evident in vivo than it is in vitro. So after an IV injection or after exposure to plasma, the adenovirus virions immediately become coated with a variety of host plasma proteins. And this forms a corona that coats the virion and actually determines its properties to a large extent in terms of tropism. So how does adenovirus type 5 bind and enter cells? So it's been known for more than a couple decades that the adenovirus fiber knob, and this is Ad5 here, but other adenoviruses bind to similar receptors or related receptors. So the adenovirus fiber knob binds to a receptor called CAR, the Coxsackie adenovirus receptor. And the adenovirus penton, shown in light blue here, binds to integrin. So it's like a two-step receptor interaction. This has been demonstrated very strongly in vitro, but it turns out if you take vectors and mutate both of these regions so that they're no longer able to bind to the receptors, the vector is able to transduce the liver perfectly fine. So these receptors are not actually necessary for hepatocyte uptake. And a breakthrough in this field was in 2008, when a number of different labs, not ours, but a number of different labs discovered that adenovirus 5 binds to coagulation factor 10. And this is critical for liver transduction. The part of the virus that binds to coagulation factor 10 are these purple Hexon trimers. And you can see there's a very large number of these Hexon trimers on the surface of the virus, 240 copies of the trimer. Factor 10, you may remember from long ago in biochemistry, Factor 10 is one of the last enzymes in the coagulation cascade. Factor 10, when activated, will cleave prothrombin to form thrombin, which then cleaves fibrinogen to make a fibrin clot. So this is very strange. Why would a virus want to bind to coagulation factor 10? This is actually a high affinity interaction, about 1 nanomolar. And it is specific for factor 10 in the sense that it's high affinity, but it's also a little bit non-specific because other coagulation factors that share structural features with coagulation factor 10 also bind to adenovirus hexon. So I'm going to tell you a little bit about some other proteins besides factor 10 that are also very important. And this is drawn to scale. So here's what factor 10 looks like next to this giant adenovirus, which has a diameter of about 90 nanometers. So there's some other proteins that bind as well, and some of them are pretty big. So there's IgM, which I'll refer to as natural antibodies. I'll tell you more about that on the next slide. There's also a variety of complement proteins of the classical pathway. And the classical pathway basically means the complement is activated by antibodies. And IgM, turns out, is a very good activator of the classical pathway. So C1q, C4, and C3 are the critical proteins, and C1q and C4 are specific for the classical pathway. So I'll tell you a little bit more about IgM now and how it recognizes adenoviruses. It turns out if you take mice that have no previous exposure to adenovirus or any other pathogen, they will have antibodies against adenovirus. So this is an ELISA assay where we've coated the plate with adenovirus and put on the mouse serum, and we can see antibodies of the IgM class binding, but not the IgG class. And this is actually fairly typical for a whole variety of viruses. There are natural antibodies that recognize these viruses, even in the absence of prior exposure. And what this actually is an innate pattern recognition system. So these IgM antibodies, because they have 10 different FAB regions, actually 10 identical FAB regions, they can actually bind to things with low affinity, but high avidity. And because of the repeating structures on the surface of adenovirus and many other viruses, these IgM antibodies are actually quite good at binding to them, again, with low affinity, but it can be quite high avidity. And it can also activate the complement system because when this binding happens, these IgM antibodies become very good at activating the classical complement pathway. Now, another thing that IgM antibodies are very important for, as I mentioned, is also uptake by Kupffer cells in the liver. So here's a very simple experiment where we've taken wild-type C57 black 6 mice and then antibody-deficient RAG knockout mice and compared the amount of uptake in Kupffer cells 10 minutes after IV injection of Ad5 vector. And you can see there's quite a lot less uptake by the Kupffer cells in the RAG knockout mice. And looking at this with a variety of other strains, we can actually look at the relationship between IgM concentration in various strains and uptake in the liver. And what I'm going to show you here is hepatocyte transduction. So we have RAG knockout mice, which have no B cells, CD19 knockout mice, which have only a few B cells, wild type mice, and then these PTPN6 B cell specific knockout mice, which actually have elevated B cells and elevated natural antibodies. And then when we look at the amount of luciferase, so this is an adenovirus vector expressing luciferase that we've injected intravenously, and compare that against the amount of serum IgM in these various mice, we can see that there's an inverse relationship between liver transduction, so hepatocyte expression, and IgM concentration. So mice that have low amounts of IgM have low amounts of Kupffer cell uptake and high hepatocyte transduction, whereas mice that have very high amounts of IgM have low hepatocyte transduction because they have higher Kupffer cell uptake. So there's sort of this balance between the Kupffer cell uptake and the hepatocyte transduction. And here's another way of showing this, but this is also an important point too. Different mice strains actually differ in the amount of IgM that they have in the circulation. C57 black 6 mice have lower IgM, and BALB/c mice have higher IgM. So here you can see the same type of relationship between liver transduction, luciferase expression, being high when there's low IgM, and low liver transduction when there's high IgM. We take F1 hybrids between these two mouse strains, they're in between. And this is actually, you can see a logarithmic axis on these on the y-axis. So this is actually quite large changes in liver transduction caused by IgM transduction, IgM concentration. So the summary so far, natural IgM is very important in affecting the biodistribution and liver transduction by Ad5 vectors. Naive mice have natural IgM antibodies that recognize Ad5. This is not specific to mice, it's also other species, including humans. And IgM inhibits liver transduction by Ad5 by increasing the uptake by Kupffer cells. So if you have more Kupffer cell uptake, less hepatocyte transduction. And this is important for preclinical studies. For example, you can imagine the choice of mouse strain could make a big difference depending, in whether your adenovirus vector works well or not, depending on what you want it to do. BALB/c mice, because they have higher IgM, will have higher Kupffer cell uptake and lower liver transduction. That may translate into lower liver toxicity. So one strain of mice may have very different biodistribution or toxicology findings than another strain. All right, switching gears a little bit to factor 10, I want to talk about how factor 10 facilitates hepatocyte transduction by ad vectors. Remember in 2008, a number of different labs discovered that factor 10 binds to Hexon and is absolutely critical for liver transduction. So here we're looking at beta-galactosidase in hepatocytes at low power. And the way this was thought to work is that factor 10 acts as a bridge receptor. So one end of factor 10, the GLA domain, is the part that binds to Hexon. The other end of factor 10 has some positively charged amino acids that form a heparin binding site, and they can bind to heparin sulfate on the surface of cells. So when you mix the virus together with factor 10, it gains the ability to bind to a new receptor, which is heparin sulfate. This is very clearly demonstrable in vitro, and it was assumed that this is also the way that it works in vivo, quite logically. But soon we found some data that was incompatible with that. So let me tell you first about evidence that factor 10 is critical for liver transduction in vivo. One way of demonstrating this is by pre-treating mice with warfarin, also known as Coumadin. This depletes factor 10 because warfarin blocks vitamin K-dependent maturation of factor 10. So you can see here when we've taken C57 black 6 mice and pre-treated them with warfarin and looked at how much luciferase expression delivered by adenovirus vector, the warfarin pre-treatment causes very poor liver transduction. And this can be reversed by pre-treating the warfarinized mice with factor 10. So we give factor 10 intravenously shortly before injecting the adenovirus, and this normalizes transduction. So warfarin prevents hepatocyte transduction, and factor 10 reverses that. This is very solid evidence that factor 10 is critical, at least in these wild-type mice. But we soon, because we were interested in natural IgM and we were using RAG knockout mice, we soon realized that this wasn't always the case. In fact, when we did these same experiments in RAG knockout mice, we got completely different results. The warfarin had no effect in the RAG knockout mice. Liver transduction is just as good with and without warfarin. And you can see that here histologically and here quantitatively. So what was going on? Another way of looking at this is to mutate adenovirus so that it no longer binds to factor 10. So instead of depleting factor 10 from the mouse, now we're going to prevent the virus from binding to factor 10 in the first place. So here's the adenovirus 5 hexon trimer, and it's made up of these hypervariable external facing residues that form sort of a cavity here, and this is where the factor 10 GLA domain binds to. And using some mutations designed by Andy Baker's lab, we can mutate the vector so that it's no longer able to bind to factor 10. And when we do that, and we look in wild-type mice and RAG knockout mice, we get very similar results. The wild-type mice, the virus that cannot bind to factor 10 is terrible at transducing the liver, whereas in rag knockout mice, it's almost as good as the wild type vector. And we also get similar results in other antibody deficient mice strains. Of course, rag knockout mice have other differences besides lack of antibodies. They also lack T cells. But when we use, I'll show you on the next slide, when we use a mouse strain that specifically lacks B cells, we get the same results. Factor 10 is not essential for liver transduction in these antibody deficient mice strains. All right, so here we're going to use a different mouse strain, JHD mice. These mice have T cells, but they lack B cells. We're going to pre-inject some of these mice with IgM to see what happens. And what effect does that have on liver transduction by the Ad5 vectors? All right, so when we take, these are all 4 groups are JHD mice that lack antibodies. When we inject IgM and use a regular Ad5 vector, there's very little effect. And when we use our Ad5 vector that is unable to bind to factor 10 and inject IgM, it is very inhibitory for liver transduction. So this tells us that the lack of IgM in these antibody deficient mice is what is the reason why they are able to transduce the liver in the absence of factor 10. To summarize that more eloquently, I have a chart here. So when antibody is present, factor 10 is necessary for Ad5 to transduce the liver. So you get good liver transduction in wild-type mice as long as factor 10 is present. But when you have factor 10 absent, you get poor liver transduction. And then when you're using mouse strains that lack antibodies, factor 10 is irrelevant. You get good liver transduction whether or not factor 10 is present, and whether it's been depleted with warfarin or whether you block factor 10 binding to the virus in the first place. Now, as I've mentioned a few times, natural IgM is very good at activating classical complement pathway. This is an amplifying enzymatic cascade, and it's triggered by a conformational change in IgM. Excuse me. When the antibody binds to something like a virus capsid, activates C1 and then C2 and C4 and then C3. And actually C3 and C4 are very interesting proteins that can covalently attach to viruses and many other types of molecules. To cut a long story short, when we do the same types of experiments in mice that lack instead of lacking antibodies, they're going to lack the components of the classical pathway. Liver transduction in warfarin-treated mice is unaffected in these C1q knockout and C4 knockout mice, where C3 knockout, it's normal. So this tells us that the classical pathway, including natural antibodies, which activate C1q, which activates C4, the classical pathway is essential. If you knock out any one of these 3 things, then you no longer need factor 10 to transduce the liver. Whereas if all of these things are present in wild-type mice, factor 10 is required to transduce the liver. So this sets up a new hypothesis. So, factor 10 gives good liver transduction in wild-type mice, and the reason was thought to be that factor 10 gives Ad5 an affinity for heparin sulfate in the liver. So certainly this is true in vitro when you're looking at cultured cells. Factor 10 is very good at enhancing transduction, and the mechanism is clearly transduction enhanced by heparin sulfate on the cell surface. So the new hypothesis in vivo is that factor 10 is actually shielding the adenovirus from attack by natural antibodies and complement. So we set up some in vitro model systems to study this further and figure out the mechanisms. Now, it turns out blood is a very difficult environment for viruses because of all of these things that I just mentioned, including natural antibodies. Many different viruses, not just adenovirus, are recognized by natural antibodies and neutralized by complement. And this is literature going back 40 years at least. However, when we did our initial experiments with adenovirus and mixed it with mouse serum, the virus is actually pretty stable in mouse serum. And that's shown here on this next slide. When we take a normal Ad5 vector, put it in media or put it in C57 black 6 serum, nothing much happens. The virus is stable. However, when we take our vector that is unable to bind to factor 10 because it's been mutated, this vector gets neutralized very efficiently when it's mixed with wild-type mouse serum. And we can do some additional experiments to look at that further. We can take XBP, which is a snake venom protein that specifically blocks factor 10 by binding to the glutamine of factor 10. When we add that to wild-type mouse serum, suddenly the virus is neutralized. And then we can also do the same experiments in rag knockout serum. Remember, these antibody-deficient mice do not neutralize, are very good at liver transduction in the absence of factor 10. So in vitro, when we take rag knockout serum, it's unable to neutralize. It doesn't matter whether or not factor 10 is present. It doesn't matter whether or not the virus can bind to factor 10. So this recapitulates what we're seeing in vivo. And what we conclude from this is factor 10 is protecting Ad5 from neutralization by mouse serum, and the neutralization depends on these natural antibodies. So let's look a little bit more at the natural antibodies, and then we'll look at the complement system. Same type of in vitro experiment here, neutralized. So this is the vector that's not able to bind to factor 10. Neutralized in the presence of wild-type serum, not neutralized with rag knockout serum. Interestingly, IgM by itself has no effect. So these natural antibodies are not neutralizing. They may bind to the virus, but they don't neutralize it. However, when you take the IgM and mix it with the rag knockout serum, neither of these things are able to neutralize on their own, but together they are able to neutralize this virus because it's not protected by factor 10. And similarly with the components of the classical complement pathway. So C1q knockout and C4 knockout mice, and to some extent C3 knockout mice, are not able to neutralize these factor 10 deficient vectors. And one thing we found about the mechanism is this adenovirus activates complement when you mix it with mouse serum, and factor 10 prevents that. All right, so here we have the amount of C3a. So C3a is the end product of complement activation from cleavage of C3. So we have cleavage of C3 being increased when we mix in a vector that's unable to bind factor 10, whereas the wild type Ad5 vector does not increase it. There's a certain amount of background levels. So the wild type does not increase it above background, but the vector that's unable to bind factor 10 activates C3a very robustly. When we add XBP, so remember this is the protein that blocks factor 10. When we add it, suddenly the control wild-type adenovirus vector is able to activate complement. So this is evidence that factor 10 binding to the adenovirus vector is preventing the adenovirus vector from activating complement. And this all depends on natural antibodies and this classical complement cascade. So this in vitro model system was very informative at telling us the mechanism, and it exactly aligns with what's happening in vivo. So just to summarize this part, we found that factor 10 is shielding adenovirus from attack by the complement system. The way it does this is by blocking the ability of the classical pathway to inhibit liver transduction by ad vectors. So that's in vivo. In vitro, the mechanism is factor 10 is inhibiting the neutralization of the vector by complement, and factor 10 is blocking the ability of the adenovirus to activate C3 convertase. So a very important question here and very mission relevant for the FDA is how far can we trust these results in mouse models? Is this really the way it's going to work in humans? This is a difficult question to answer for a couple of reasons. First of all, we can't do the in vivo experiments in humans. But the other reason it's difficult is that humans, unlike our mice, humans will have been exposed to adenoviruses typically at some point in their history, and will have different types of antibodies. So IgG antibodies, many of those can neutralize on their own. They can also activate complement. And this varied adenovirus exposure history among humans makes it very difficult to answer these types of questions. Nevertheless, we were able to investigate it and answer it. So here's an in vitro neutralization assay. I'm just showing you the results with mice. They're the same as what we had on the other slides, except this is a log scale here now. So when we take our vector wild type at 5.0, non-factor 10 binding at 5.7, mix them with media, no difference between them. Mix them with mouse serum. The vector that's unable to bind factor 10 gets neutralized. The other vector is fine. All right, so factor 10 in mouse serum is preventing neutralization. When you don't have factor 10, the vector gets neutralized. So what happens in human serum? Well, we screened a very large number of human sera. And of course, as I just mentioned, there's a variety of pre-existing antibodies. So sometimes these antibodies are able to neutralize. So all these black bars, even in the absence of this mechanism, they're able to bind and neutralize. So even if, for example, if you kill the classical complement pathway, you still get neutralization. But nevertheless, every single human donor had a big difference between neutralization with a virus that can bind factor 10 in the black bars versus a virus that can't bind factor 10 in the gray bars. So this is evidence that it works the same way in humans as it does in mice. Factor 10, human factor 10 in human plasma is preventing neutralization of Ad5, just like in mouse serum. All right, so we've expanded our studies recently beyond adenovirus 5 to other ad serotypes. And as I mentioned at the beginning of the talk, this follows a trend in the field. Still most of the adenovirus vectors in clinical trials are based on adenovirus type 5 for historical reasons. But there's an increasing number of vectors being used for both gene therapy and oncolytic vectors and vaccines as well, using other serotypes. Some of those other serotypes have advantages in terms of tropism for different types of tissues or less seroprevalence. So they may make better vectors because there's less history of exposure in the human population. And then we're also expanding our studies beyond factor 10 to other coagulation factors. And remember, I mentioned there's some other coagulation factors that have a similar structure as factor 10. So I'm going to tell you a little bit more, particularly about expanding to other coagulation factors, but about both of these issues. Now, there's a very wide diversity of adenoviruses, not only in humans, but in other species as well. So here I'm showing a subset of the human adenoviruses, and Ad5 is in this species C, and that consists of Ad5 plus 1, 2, 6, 57, and 89. 89 is a relatively new serotype that is, Hexon is almost identical to Ad2. And then we have a variety of other adenoviruses. Most of them are in species D, so not all of them are shown here. And they cause a variety of different diseases. And part of the reason for these different diseases is different receptor binding and tropism. Anyway, I don't have time to tell you everything that we've been doing. We've been working on Ad48, and we published some on that. We're also working on Ad11 and Ad35 more recently. But what I'm going to tell you about today is our work on these species C viruses that are highly related to Ad5. So we thought it'd be interesting to look at the most closely related adenovirus serotypes and see whether we're seeing the similar impact of factor 10 and antibodies and complement as we are with Ad5. And another advantage of using these closely related vectors is it's possible to make hexon swaps between them. So this isn't possible with all adenovirus serotypes, but with closely related ones, it tends to work pretty well. So we can take Ad5 vector and keeping all of the proteins the same except the hexon, we just switch the hexon and make vectors that have Ad1 or Ad6 or Ad57 hexon. And then we can study those both in vitro and in vivo and see how they're the same or different as Ad5 vectors. And then the coagulation factors. So this is the structure of factor 10. The GLA domain is the part that binds to hexon. And as mentioned, there's a number of other coagulation factors, and some of these have already been studied. And factor 10, factor 9, and factor 7 are all able to bind to adenovirus hexon. And interestingly, factor 2 has never really been looked at. So this is prothrombin. Prothrombin is, one step further than factor 10 in the coagulation cascade. And the reason we thought this was particularly interesting, first of all, it's got a slightly different structure than the other ones, although it still has the same type of GLA domain. But factor 2, or prothrombin, is the most abundant coagulation factor in plasma. It's 10 times more abundant in terms of micrograms per mil than factor 10. And some of the others that bind to adenovirus, especially factor 7, really tiny amounts of that in plasma. So we thought because of the conservation of the gliodomains and the high concentration of factor 2 in the plasma, this would be an interesting further step to look at. And indeed, we found that factor 2 binds to a variety of adenoviruses, and there's some interesting differences between the viruses and interesting differences between human and mouse. So here on the left, we have humans, human factor 2, and here on the right, we have mouse coagulation factors, mouse factor 2. And upper is going to be higher affinity, smaller KD. Lower will be lower affinity. And we have our, I think this is wild type AD1, 2, 5, and 6, and our pseudotyped AD57 vector. And you can see there's a interesting pattern here where AD5 has lower factor 2 binding, and the other ones have higher factor 2 binding. This is similar between humans and mice. And when we look at factor 10 binding, so here, human factor 10 binding, there's actually an inverse pattern. AD5 binds well to human factor 10, but the others not so well. And the pattern with mouse factor 10, interestingly, is different. All 5 of these viruses bind very strongly with nanomolar affinity to mouse factor 10. All right, so you can see there's differences among the AD species C viruses, even though they're all highly related. And there's differences also between humans and mice. And these are driven by sequence differences in the proteins. So sequence differences in Hexon are driving these differences between the viruses, and sequence differences in the gliodomains of the coagulation factors are driving the differences between human and mice. So just very briefly before I wrap up, I'm going to tell you a little bit of work we've been doing on one of the closely related viruses, AD6. And it turns out that AD6 vectors are less dependent on coagulation factors for liver transduction. And they also have some interesting properties in being less uptake by Kupffer cells as well. And I'm not going to tell you about that today, but what we've done is made a series of vectors. So this is our AD5 pseudotyped with the AD6 hexon. And then we've made a variety of point mutations in AD6 hexon in the same vector background and looked at how that affected liver transduction, shown here, and coagulation factor binding, shown here. So we're, since we're doing these experiments in mice, I'm showing the mouse coagulation factor binding data here also. But we've also done this with human coagulation factors and also bovine coagulation factors. And so you can see we were successful in making a variety of mutations in Hexon that greatly decreased coagulation factor binding. However, when we tested these vectors in C57 black 6 mice, they all had very similar liver transduction. So whereas if you do this experiment with an AD5 vector, it can almost completely knock out liver transduction. With AD6 vectors, they don't seem to matter so much. We've also done these experiments with warfarin pretreatment, and the AD5 vector is extremely dependent, extremely suppressed by pretreatment with warfarin, whereas AD6 vectors or AD6 pseudotype vectors, shown here, are not suppressed so much by warfarin pretreatment. So AD6 vectors are less dependent on coagulation factors for liver transduction than AD5 vectors. So this is a very interesting finding. All right, so I'm going to wrap and then we'll take some questions. But what I've told you today is our work on adenovirus gene therapy with non-replicating vectors and how this is much more complicated in vivo than in vitro. And this is to a large extent because of the interaction of these plasma proteins, including coagulation factors, natural antibodies, and complement. You can see they interact with the virus in different ways. They also interact with each other and influence each other in complicated ways. And both in vitro and in vivo studies are necessary to figure that out. We've shown that factor 10 binds to AD5 and protects from natural antibodies and complement. And this is actually the mechanism for how factor 10 enhances liver transduction by AD5 vectors. In contrast, other adenovirus serotypes, even closely related ones like AD2 and AD6, bind factor 2 more strongly than factor 10. And again, we think this is important because factor 2 is much more abundant in the plasma than factor 10 is. And then just a general theme in our work is that it's very important to pay attention to species-specific differences between animal models and humans. After all, the goal is to find out what's going to happen in a human, not what happens in a mouse. So for AD5 at least, the mouse is an effective model for this aspect of biology. As I showed you, with a little bit of data, we have more extensive studies that we've already published, that the human natural antibodies and complement and factor 10 all seem to work the same way as the mouse corresponding proteins do, at least for AD5. This is not going to be true of other AD serotypes. So this is important to keep in mind as you're planning preclinical studies. And then I'll just close there by thanking all of the members of my lab. We have a very small lab, but a large number of people that have contributed to this work over the past 20 years or so. And I particularly like to single out Zhili Zhu and Ji Qian, who have been in my lab for a very long time and did a large portion of this work. And then I'll show you some contact information here, both for me personally and for interesting and important resources at the FDA. And I'll be happy to take your questions now. And thank you for your attention.
Thank you so much. Thank you, Andrew. So for those who would like to ask some questions, please, you can raise your hand. You can type them in the chat and I can read them for Dr. Byrnes. So far we have 2 questions in our chat. The first one is from Dr. Masato Yamamoto. What is the target cell when you discuss the neutralization? Hepatocytes?
Yeah, so in vivo, I assume the question is about in vivo. So in vivo, what we're looking at is the amount of luciferase or beta-galactosidase that's being produced after injecting a vector intravenously. And that's almost entirely due to hepatocytes. And so that's what we're essentially looking at. We're not isolating hepatocytes. We're just taking the entire liver and mushing it up and looking at the amount of protein being expressed. In vitro, we've done a variety of different cell types, including hepatocytes, including a variety of cancer lines, and we get very similar results with all of those cell lines.
The second question, what killed Jess Gelsinger?
Oh, well, that's still a matter of debate. So this is just for those who are not familiar with this, in the late 90s, there was a clinical trial at the University of Pennsylvania for ornithine transcarbamylase deficiency. And Jim Wilson's group was using an AD5 vector that expressed ornithine transcarbamylase. And they were giving pretty high doses. And one of the subjects in this study, Jesse Gelsinger, had a severe adverse reaction that seems to be related to liver toxicity. And it's still unclear, I think, whether it's something to do with the disease, because that disease in particular is very variable from patient to patient, or it may have had something to do with pre-existing immunity. I know Jim Wilson published a paper a few years ago about pre-existing antibodies in that patient and how they were different than other patients. So that's a very interesting and plausible hypothesis, but I think in the end, that may remain unknown. And this has had an unfortunate damping effect on the field. Almost all of the clinical trials being used that use adenovirus vectors are using, even today, intratumoral injection rather than systemic injection. And this is in part because of that legacy, but also because of the problems I mentioned throughout the talk about the, how difficult it is to prevent liver uptake and liver toxicity from these vectors. So I think even more than 20 years after that incident, there's still clearly room for improvement in adenovirus vectors, although we all think they're extremely promising. There's still aspects of it that could be further improved to make these vectors more safe and more efficient.
So one more question from George Aslanidi. Is there a difference in neutralization depending on organ target in liver versus muscle?
Yes, so that's a good question. I assume you're meaning intravenous injection, the way we do it, versus intramuscular injection. And if you're injecting locally into a tissue, the amount of these proteins that are going to be around the antibodies and the complement proteins are going to be much lower than if you're injecting systemically. So this applies for intramuscular injection, intratumoral injection as well. The plasma proteins are going to be much less effective at neutralizing the virus. So this is a good thing for those routes of injection. They'll be less susceptible to neutralizing antibodies and also these mechanisms as well.
So, and I have a few questions. I don't see raised hands. I'm not sure if Sandra is able to unmute those who would like to answer the questions directly. So, but I can ask my question. First of all, yes, we just, every time when we discuss adenovirus-based therapeutics, someone concerned would be related to this tragedy which happened with Jess Gelsinger. So, and I believe that the history of adenovirus-based therapeutics with all these achievements and clinical failures did actually, yes, it raised a lot of questions about the safety of adenovirus-based therapeutics. And very often those failures are widely publicized and accompanied by negativism from both public and scientific community. Do you think that the recent success in clinical trials and the fact that most of virus-based vaccines against COVID are based on adenovirus will help the clinical translation of other adenovirus-based therapeutics? What is your insight on the future of adenovirus as the vector for vaccine development, anti-cancer drug development?
Well, of course, like you, I'm going to be very enthusiastic about that future because I've spent my whole career working on adenoviruses, and I see how versatile they are, and also how some therapies can actually use these things, these aspects of adenovirus that may be disadvantageous in some context, use them to their advantage. So for cancer therapy or for vaccines, a certain amount of innate immune response or T cell immune response induced by the vector could actually be a good thing in terms of causing, in this case, beneficial immune responses. So I think, if you look at the history of other vectors as well, so as you're all aware, AAV vectors are extremely promising for gene therapy these days, but there have been a number of deaths and serious adverse events in those clinical trials fairly recently. And this is something that, FDA is concerned about, and we had an advisory committee meeting fairly recently on that topic. And that has not, knocked out that field because AAV vectors are so promising. And I think it's the same thing with adenovirus vectors. People have continued to work on them and develop them further. As some, hopefully some products show success and get licensed, that will increase the amount of interest in developing these further. Venture capitalists and companies always like to see a successful example in order to make them feel more confident about putting their money into these types of things. So I think that's largely what's going on, and it'll be a matter of time before some of these adenovirus-based therapies are successful and licensed. And
another question: any insights on the use of other models? As a pig model to study adenovirus by distribution, as we will discuss later today. So the biodistribution of adenovirus-based vectors in pigs is quite different from what we see in pigs. At least it doesn't target the liver, it's mostly distributed in spleen and lungs. Any insight on what's going on in pigs compared to the rodent models?
Yeah, so of course, Julia has developed a wonderful pig model, and that has some advantages. Adenoviruses tend to be fairly species specific. So if you're, and I didn't mention this, but if you use wild type adenovirus 5 in mice, it actually doesn't replicate that well in mouse cells. And pigs, it does replicate better in pig cells. And then pigs are also a good model in the sense that they're a large animal model, and they can be used. They're commonly used in some types of applications like cardiac gene therapy. There's well-accepted pig models, so it might be useful for other things, including cancer, but also other things as well. So we have not studied pigs, and I should mention there's also a couple of other animal model systems that are widely used, Syrian hamsters and cotton rats. And part of the reason for using those is adenoviruses, human adenoviruses do have some extent, some ability to replicate in those animal models as well. The biodistribution to the lung is interesting because the reticuloendothelial system, which is the set of intravascular macrophages, is different in certain animals than it is in humans and mice. And pigs have these macrophages called pulmonary intravascular macrophages, which are actually in the pulmonary capillaries. And they clear particles from the circulation in the same way that Kupffer cells clear particles from the circulation in the liver. So humans normally do not have pulmonary intravascular macrophages, nor do mice. But in certain pathological conditions, they actually develop these pulmonary intravascular macrophages. And early on, and I didn't talk about this today, but we set up a model of liver cirrhosis, in part because we were interested in what happened to Jesse Gelsinger. He didn't have liver cirrhosis, but we were interested in looking at the effect that pre-existing liver damage might have on the toxicity of adenovirus vectors. And we found that animals with liver cirrhosis, the biodistribution of the vector was actually completely different. And it was because of these pulmonary intravascular macrophages that develop in the lungs in liver cirrhosis. So you could say that the pig is actually, more similar to a human that has liver cirrhosis than to normal human. And that doesn't mean that the pig is not going to be a good model system for normal human. But you need to take these species-specific differences into account. There's also some interesting data from Schering-Plough a long time ago when they were developing their AdP53 gene therapy, and this is all published. But they did some preclinical studies in pigs administering it intravenously, and a number of the pigs actually developed shock shortly after receiving the adenovirus vector, and this may have been due to a similar mechanism that I mentioned that we studied later in our shock model in rats.
Mm-hmm. So there is one more question from George Slanidi. So, but I'm not sure how much time we have. So, but I would like to remind everyone, especially for those who are interested in a group meeting. So actually, Dr. Andrew Byrnes was kind enough to suggest the group meeting for those who are interested in their career with the FDA. And we will have this meeting at 2 p.m. Please let me know if you're interested, and I will send you the Google invite so you can join this group meeting. Again, that's for those postdocs and trainees and PhD students who are interested in their career with the FDA. So, and the very last question from George, how modifications on adenovirus make difference in scale-up production? So, and I believe that's the last question.
Yeah, that's a good question. So some of these changes that I was mentioning today are on these surface exposed loops of Hexon. And as I showed you, Hexon actually makes up a very large portion of the surface of the virus. So you can imagine how changing those might affect things like affinity chromatography or anion exchange chromatography by changing the charge of the virus. So they can definitely have an effect on methods for purification. So they might require some adjustment. In terms of scale up, the types of modifications that we made have no effect on the replication ability of the virus. So I wouldn't expect some differences there, but some other types of modifications certainly could affect that.
Okay, so thank you so much. I think, and I guess we had more questions than that, but I was not unable to unmute those who wanted to ask those questions in person directly. So, and Sandra, I think she's not here, but thank you so much. I would like to thank you again, Andrew, for this wonderful seminar. And I'm looking forward to have more meetings with you later today. Thank you so much.
It was my pleasure. Thank you, everybody.